the scope and scale of your current work in your laboratory.
Okay. One thing we do, and that is the crux of our work for downstream applications, is we collect a biorepository of human donor eyes, fresh human donor eyes. And we do this in collaboration with the Utah Lions Eye Bank and the people in Utah and the Salt Lake City community. And we get the utmost support from our chair, Dr. Randy Olson. And we work in very close relationship with the clinicians, the retina specialists in our department, and also Ivana Kim at Massachusetts Eye and Ear Infirmary for post-mortem phenotyping. And that is very critical to make sure we have the right phenotype or the right diagnosis of age-related macular degeneration. Because if the eyes are not properly diagnosed, then you cannot trust your results from your science. So that is what we start with. And we have a wonderful laboratory of postdoctoral fellows, graduate students, and technicians to process the eye tissues, because many of them come in the middle of the night, 2 or 3 o'clock in the morning, because you can't anticipate when loved ones may pass away. And this goes on 7 days a week, 24 hours a day, every third week of the month. And we get many eyes, and we're on schedule to get 1,000 donors or 2,000 eyes this year alone, which is significant. But it costs a lot of money to do that, and it costs a lot of money to process those eyes. So we apply for grants to do that, and we apply for grants to study those eyes. And we feel that is the best way to get to disease mechanisms and develop appropriate therapeutic targets. Because while animals are good, they simply don't recapitulate or duplicate what is going on in the human eye. So.
Tell us about some of the work you're doing with these eyes.
Some of the work we're doing with these eyes is we're not only looking at the genetics, but trying to study what is going on in the environment. And that is the field known as epigenetics. So things that your parents do, you can inherit. And what I mean by that, if your parents smoke, or the nutrition they take through their lifetime, or if they drink alcohol and medications they may take, you can inherit that. And things that you do through your lifetime, how you live a healthy lifestyle, whether you exercise, how you eat. If you smoke, if you're exposed to toxins in the environment, that can affect your genome. And it can mean whether you're going to have a risk towards age-related macular degeneration or whether you might be protected from age-related macular degeneration. Those are the things we're trying to study in the genome, and we're using these donor eyes to study that.
Tell us about some of the research that you presented here at ARVO 2016.
And that is exactly what we presented on Saturday at the Epigentics Symposium on Saturday. And we presented some of the results for that. And we showed that the lipid pathway, or fatty acids cholesterol pathway, which other researchers like Christine Curcio, Jerry Luddy, Steve Fleisler have shown that that's an important pathway in age-related macular degeneration. Dimitrios Favis, Joan Miller have shown, also Robin Geimer, Paul Baird. But we've shown for the first time that epigenetics and allele-specific expression, that lipid biosynthesis is also important. And we've shown some novel genes that haven't yet been shown that are also important, and those may represent new therapeutic targets. And we're quickly in the process of trying to submit that paper, and your organization has generously supported that research, so we've been very lucky.
Where in the lipid biosynthesis pathway do you think the impact is being made?
In cholesterol. specifically targeting cholesterol. How that is happening, we still needs to be teased apart. And those are going to require benchtop experiments. And those assays we're still trying to develop as we speak. They're going on in our laboratory.
So that turns to the next milestones in your research. What are your goals for the next year?
The next year is our postdoctoral fellowship. Just got a training grant sponsored by the National Eye Institute, Katie Pennington, and she will actually be working on those assays and trying to figure out allele-specific expression. So we expect that we inherit one allele from our mother, one allele from our father. And we expect those alleles or those gene loci to be expressed equally. But sometimes they're not. One preferentially expressed. And we want to know why that is, because that could explain, due to regulatory mechanisms, why the disease shows differently. And we want to take advantage of why those alleles are expressed differently. So we're going to test those assays. We're going to test for that in the actual eye tissue, whether it be the retina or the retina pigment epithelium, and compare that between disease states.
What should the general public and family members and individuals with age-related macular degeneration take away from the current state of your research?
To please get involved in many studies as you can, and to really thank them and appreciate for what they're doing by donating their organs of their loved ones. And we really are grateful for everything they do, because without them and their donating eye organs to the donor eye repository, especially for Utah, we could not do the work that we do without them. And just a lot of thanks and gratitude.
Professor, is there anything else you'd like to add about your programs, your trajectory, or other issues?
Besides the eyes they give of their blood also. And that's important because we do not know if this is a localized or systemic disease, or if there's certain aspects of the disease that could be localized or systemic. So when someone comes and asks you for a sample of blood or to donate your eyes upon death, please think twice about it. You know, I'm an organ donor. My mother and my aunt, who are both deceased, are also part of my study, and they have the disease, and I hope to cure this disease in my lifetime. And it's people like your organization and people who participate in our study that really drive the research and really enable it to move forward. And we really are grateful to that. Thank you.